it's my pleasure to introduce I'm going to call her a new Canadian, Kanta Subbarao. Professor Subbarao holds now a Canada Excellence Research Chair, joined the University of Laval in Quebec City in the Department of Microbiology and Immunology. She's an honorary professor in the Department of Microbiology and Immunology at the University of Melbourne, at the Peter Doherty Institute, and she's a physician scientist whose research over the years has focused on newly emerging and re-emerging viral diseases of global importance, including pandemic, seasonal influenza, and coronaviruses. She is a friend of Dr. Kang, because they both trained and did their medical degrees at Christian Medical College in India. And following her training there, she went to the US and completed her residency and fellowship in pediatric infectious diseases. And then over the years, she has moved on. She went to the NIH, and she was the chief of the molecular genetics section of the influenza branch, then at the CDC from 1997 to 2002, chief of the emerging viruses section at the Laboratory of Infectious Diseases at the NIH, and then director of the WHO Collaborating Centre for Influenza in Melbourne until this year. And it's at that point that Canada managed to persuade her here. So we're very happy that she's here. And I would like to introduce her and ask her to take the stage.
Well, thank you very much for this invitation. As Janet said, I'm new to Canada, but very happy to be here. And I'm going to talk to you about respiratory viruses, which I have worked on for many, many years. The range of viruses, they're viruses that are endemic in humans as well as in animals. We often see spillovers, as you heard from Malik, of viruses going from animal hosts into humans. And then we have the occasional pandemics, as we've just come through one. And anyone in the flu field will tell you that it's inevitable that we will have another. So these are the three versions that respiratory viruses exist in. And the ones that we fear most are pandemic influenza viruses or pandemic respiratory viruses. So there's no definition of a pandemic virus, but there are features that are shared. And the features are that it requires the isolation of a virus that is novel to humans, to which the general population has little or no immunity. The virus must infect, replicate, and cause disease in humans, and very importantly, must spread efficiently from person to person in sustained chains of community-wide transmission. So as you heard from Malik earlier, we were all surprised when SARS-CoV-2 emerged in early 2020. But I remember hearing on February 1st the two features of SARS-CoV-2 that differed from SARS-1, and that was spread from an asymptomatic individual and spread from efficient spread from person to person. So those two features really accounted for the epidemiological success of SARS-CoV-2. So how do we now distill down from the universe of viruses that are present in animal species or those that have circulated previously in humans to figure out which of those have pandemic potential? So so I'm going to take a very much more virologic approach to this question, because you've heard from Malik and Jane about the other aspects of how you control pandemics. So from the virologic standpoint, how do we know what's going to infect and cause disease in people? So the virus has to find its cognate receptor. So the receptor has to be present on human cells, and the virus then has to be able to replicate in these cells and evade innate immunity. So we know now, for many of the coronaviruses and influenza viruses, we know what the receptor is. There are subtleties around those receptors, but we can look for the presence of receptors, which is the first factor that has to occur. The virus actually has to find its receptor, attach, and enter into the cell. It also has to evade the innate immune response, and there we can have methods to sort of look at that. So what we have done in my lab, and there are other labs that do similar work, is that we've learned from the COVID-19 pandemic that it wasn't really super helpful to know whether a virus grows in African green monkey kidney cells, in vero cells. Most virologists are really excited when there's a new pathogen and we get it to grow in the lab, because we then can work with it. However, what we learned during COVID-19 was that what we saw when we looked at infection in vero cells was quite different from what we saw when the virus infected different parts of the human respiratory tract. So what we've done is to try to reproduce the human respiratory tract in an in vitro system. And we do this in multiple ways, starting with the nose, so you can get nasal scrapings and you can grow those cells at an air-liquid interface. Let me go forward one. At the air-liquid interface, these cells can differentiate. It takes about 28 days for this to happen. And you do this by growing the cells on a permeable membrane, and you could put the nutrients below. When you expose the cells to air, they differentiate, and you get this multi-layered, and you get the ciliated cells at the top. So we've done this now, so nasal cells, and then you can get airway cells from the large airway or the small airway. You can get these from bronchoscopy samples, or you can buy them from companies. We had to find a way around this because the companies wouldn't ship to Australia during the pandemic. So we had to find alternative ways of getting these cells. And Ron Crystal from Cornell immortalize the basal cells from bronchoscopy samples, and you can actually freeze them down and revive them and differentiate them. So we're less dependent on bronchoscopies. And then for the alveolar type 2 cells, we can use pluripotent stem cells that can be differentiated into alveolar type 2 cells. And that gives us what's happening at the alveoli. In all of this, you're missing the architecture. And so that's been one of the criticisms. And so we've now started doing precision cut lung slices, either from animals or from humans, where we can actually cut a punch of live tissue, slice it, and put each slice into a well of a tissue culture plate, and they can survive for 2 weeks. So you can infect the cells of the lung in the context of its neighbors. Of course, we're still missing the immune system, and there are people that are looking at ways to bring the immune system into this. So again, the large and small airway cells also differentiate, and you can see from these images that they are different, that they differ from each other. So it's important to be able to reproduce this. So we can then take, these are seasonal human coronaviruses, and there are 4 that are currently known: NL63, 2290, OC43, and HKU1. So the one that's missing here is HKU1 because it's very hard to work with in the lab. It only replicates in airway cells. But this is an example of putting each of these human coronaviruses into all 4 of our cell types. And you could see that OC43 only replicated in our hands in the lung AT2 cells, whereas 2290 and NL63 replicated in different cells. So we'd like to use this as a system to say, if you've got a novel virus, if we've got a virus that we're concerned about its pandemic potential, when we put it into the different cell types, does the virus replicate? So this is an example where we've taken human large airway cells on the left and ferret precision cut lung slices on the right. And we've taken highly pathogenic avian influenza viruses and we've put them into both of these systems. And you can see that both of these viruses, a 2005 H5 virus and an H5N6 from 2014, replicate in both systems. So we believe that that is one way to take the universe of viruses and say how much of a pandemic threat do they pose. The second important question is whether the population is susceptible. So for this, we ask several questions, and we go through this exercise or activity every time a novel virus emerges. We did this in 2009. So the question is a segment of the population immune or partially immune? Are there serologic assays? Sometimes you have to develop them from scratch, and then when you're developing these assays from scratch, you have to figure out whether you have the appropriate controls and whether you can interpret the data. Cellular immunity assays are much more challenging than serologic assays, but it's been very clear during the COVID-19 pandemic that we really need to get a handle on doing cellular immune studies. And then, of course, interpretation of the immunologic data. This will help us understand who's most susceptible, and in the event that we have prevention and control strategies, how do you target them? So this is an example from 2009 when the swine origin influenza H1N1 virus emerged. And these are data from the CDC where they happened to have sera from each decade of life that they then tested against the newly emerged virus. And what they found was that people born prior to 1950 had some antibody. And this, in fact, explained why the older adults in the world were spared from the 2009 pandemic. So this question comes up each time, and so it's important to set up biobanking and set up serum banks so that we can go back and look at who is most susceptible. So the third element is the ability of the virus to spread efficiently from person to person. So there are a number of, and this goes back to when you think about where we were at the beginning of the pandemic in 2020, where we were talking about what the mechanisms were by which the virus SARS-CoV-2 was spreading. And so there certainly is, so people exhale the virus. It can actually drop onto surfaces, as you heard from the previous speaker, but the small particles can remain suspended in the air in aerosols. And so it turns out we had a lot of focus on wiping down our groceries and endlessly hand washing, but the reality is that airborne transmission is what really leads to extensive spread. So I was fortunate enough to go on a safari in South Africa, and this is just a reminder of how important sneezes are. So there are virologic traits that determine how well a virus could spread through the air. So we know, for instance, that there are changes in the hemagglutinin. There is the match between the hemagglutinin and neuraminidase, fusion characteristics, as well as the polymerase gene that can make virologic determinants of what might be successful in spreading through the air. But in addition, in the lab, we use the ferret model to look at airborne transmission. So when we have a novel influenza virus, and we could see several examples here of viruses that our field characterizes, and we use a ferret model because unlike mice, ferrets actually get sick with flu, they cough and they sneeze, and the virus can spread through the air. So you can set up a system where you experimentally infect one ferret and introduce a naive ferret into a adjoining space separated by a mesh so they can't touch each other, but the air can cross. So usually when you have a novel virus that emerges, one or more people in our field will put the virus into an airborne transmission model and say, well, this virus spread, and therefore it ratchets up the pandemic potential. And it's usually a Science or Nature paper very quickly claiming that this is going to be the next big threat. But we found that there were actually a few occasions where the ferret model overcalled the the airborne threat. And two examples are H7N9 and H3N8 that did transmit in our ferret system, but they didn't transmit in humans from person to person. So we wondered whether there was a way to modify the ferret transmission system to say, well, maybe putting 6 logs of virus into the nose of a ferret and then seeing if it transmits is not totally fair. Maybe what we need to look for is sustained transmission. So we did this very large experiment where we looked at whether the virus that we experimentally inoculated with, when that spread to a neighboring ferret, we would then take that ferret out and make that the donor. So we were now no longer flooding that ferret's respiratory tract. We were just saying, could sustained transmission occur? And it turned out, to my surprise, I was expecting that the viruses that didn't spread in people would not spread in a sustained manner in ferrets. But it turned out that once we were able to infect a ferret, they did transmit. So we believe that the transmission in the ferret model is very informative for when it doesn't transmit. But when it transmits in ferrets, that is really necessary but not sufficient to explain whether the virus would transmit in humans. So this is a space we need to continue to work on. And I've become very interested in studying airborne transmission. We now have new technologies where we can actually recover infectious virus from the air. And we're doing this in COVID-19. In the lab, we can do it with influenza. We can nebulize influenza, and we can collect the air, and we can recover infectious virus, not just the RNA. So we are able to distinguish between what the amount of infectious virus. But we really need to work with epidemiologists, infection control physicians, mathematical modelers, engineers, and aerosol biologists, because one of the things that I have learned is that what I was taught in medical school about the magic number of the particle size and distance is not actually true. Aerosol biologists will tell you that even in normal tidal breathing, we produce aerosols. The aerosols are about 50 to 100 microns in size, and they rapidly decrease in size with evaporation. So this is an area that is, we're coming together, fields are coming together, talking, trying to establish some links and rethink some of our definitions. So in that first set of features that I told you about pandemic respiratory viruses, the part that was missing was severity. And the WHO was actually criticized quite a bit for declaring the 2009 virus as a pandemic because it didn't cause very severe disease in many parts of the population. So they've gone back to look at severity and to try to collect indicators of severity around the world. And they've encouraged each country to develop estimates of burden of disease so that we can incorporate that as well into our discussions. And so there is an effort to try to define influenza severity. So what happens when you have a novel virus, or we're preparing now for the appearance of a novel virus? So we have to think about transmission, about virulence, as I said, in terms of severity, and medical countermeasures, which is the MCMs. So these are some of the features that we need to find a way to triangulate to try to narrow down the list of pathogens that we worry about. So the WHO, as have other organizations, so NIH has been through a similar effort, they have gone through a prioritization of different virus families to say which virus families should we focus on, which virus families represent a high, medium, or low pandemic threat. So they've just gone through this exercise, and this report has just been published. And you can see that there are a huge number of RNA viruses that we worry about, there are bacteria that we worry about, and the pox viruses on the DNA virus side. And they've also broken it down by part of the world. And I will point out to you, as an influenza virologist, that the map that is red hot across the world are orthomyxoviruses, or influenza viruses. So there is consensus that we worry most globally about influenza viruses. And so this is an effort to sort of look at it in terms of how do you select what to focus on, because the universe of pathogens is really large. And then once you focus on a group of pathogens, you have to develop a risk assessment strategy. So there are a couple of these that have been done. The CDC started off with something called the IRAT, the risk assessment, influenza risk assessment tool, and the WHO has modified that somewhat to what's called TIPRA, and it is the tool for influenza pandemic risk assessment. So in version 1 of TIPRA, it was triggered by one human infection. So if there hadn't been a zoonotic infection, TIPRA wasn't triggered. But version 2 now recognizes that we probably need to pay attention to what's happening in the animal world as well. So if there are any other animal influenza viruses that may not have caused human disease, but have some of the features that we worry about, that we should pay attention to them. And so what we do in the TIPRA system is look at a hazard assessment, an exposure assessment, and context assessment to characterize the risk. And so TIPRA uses 10 risk elements. They look at the several properties of the virus, and some of this will be what I've just told you about, the receptor binding characteristics of the virus, genomic characteristics of the virus, the transmission in animal models, and susceptibility to antiviral treatment. We look at attributes in the human population, whether human infections have occurred, what the severity of disease is in the cases that have occurred, what is the population immunity, and that has two facets. It determines how susceptible people are, that is, that there's a likelihood of a pandemic, but also the impact of a pandemic. So how are there segments of the population that are already immune? And then virus ecology and epidemiology in non-human hosts. What is the geographic distribution in animals, and have infections in animals occurred? So this is the way TIPRA's output comes out. So they invite a group of experts from many different fields. People score information on two outcomes. What is the likelihood that a virus would emerge to cause a pandemic? And if it emerges, what is the likely impact? And that's what you triangulate to place the dot. And so this is just a hypothetical of the likelihood and impact of a virus if it acquired the capacity to transmit from person to person. So when do we do this? We ask what is the likelihood of sustained transmission and the impact, as I said. So we do this when there's a rapid geographic spread of a virus, when there's an increase in the number of host species infected, and Malik showed you the extensive change in how avian influenza high-path H5 viruses have infected so many species, aquatic species through the Americas, and spread in non-mammalian hosts, especially pigs. The virologic triggers, I mean, we can sequence gaze, and we can identify, we know certain amino acid hotspots, we know what might determine receptor specificity changes. We know some of the features that link to an increase in virulence or transmissibility. We can look for molecular markers of antiviral drug resistance in the case of influenza, and more so now gradually with the coronaviruses, and then reassortment or recombination between animal and human viruses. So there are all these triggers that might actually make us assess a zoonotic virus. So what do we do once in addition to the risk assessment, how do we progress from these selected prototype pathogens? This is a slide that was originally made by the NIH, but has also been used in the WHO exercise. And it essentially tells us that we need to conduct research and develop antiviral and vaccine strategies, and then progress them through phase 2. So we've been surprised twice recently, or 3 times in this century. We were surprised when the 2009 influenza pandemic strain came out of pigs rather than out of birds. We were surprised when the SARS-CoV-2 emerged. I think everybody in the flu world was expecting the next pandemic to be an influenza virus, and it wasn't. It was a coronavirus. And as Malik showed you, we've also been surprised by the emergence of H5N1 high-path avian influenza in dairy cattle in the US. So I have just arrived in Canada a few months ago, and it is to set up I have a Canada Excellence Research Chair position to set up a center on biology and control of zoonotic and pandemic respiratory viruses. The plan is to sort of look at what's in animals, look at the animal-human interface, susceptibility, and transmissibility. And this is beautiful Quebec City. So as I said, we were surprised by what happened in dairy cattle this year in the United States. The illness in dairy cows appears to be primarily a mastitis, and there's very, very high titers of virus in the milk. This is the latest situational update covering the entire period, and it started, as Malik said, as a single transmission from an avian species into cattle, but then has spread through 14 states. And at this point, there are 27 reported or likely human cases. So is that the tip of the iceberg? Are there more out there? I think every time an animal pathogen crosses into a mammalian species, there is the risk that it will acquire the properties that will enable it to adapt to the mammalian species and spread. So these are the things that are concerning, which is why I think we're here talking about pandemic preparedness. So thank you very much for your attention.